everybody. Thank you for coming to this university lecture. My name is Mike Mazinski. I'm a professor in the Department of Genetics, Development, Cell Biology, and we're quite happy to have Kevin Folta here tonight to speak with us. So Kevin got his PhD at the University of Illinois Chicago with Lon Kaufman and did his postdoctoral work at the University of Wisconsin-Madison with Edgar Spalding, both in the areas of photomorphogenesis. After that, he assumed a faculty position at the University of Florida, Gainesville, and has been there ever since, working on both photomorphogenesis and also in the area of functional genomics of small fruit flavoring. He's also had quite a number of efforts in mentoring everyone from high school students through undergraduates, graduate students, and postdocs through his work at the university, where he is now professor and chair in the Department of Horticultural Sciences. He also has quite a bit of effort in public outreach, especially trying to help scientists communicate to the general public what we do and why we do what we do. And in that vein, he's actually been quite active in the past 10 years in communicating with the general public on many different topics, including water use and water sustainability, climate, vaccines, and GMO issues. And so tonight, he will talk to us about GMO technology and the future of food. So thank you, Kevin.
Okay, thank you very much, Mike. Wow, everybody, thank you for coming out. That's very nice. I really appreciate the opportunity to talk to you tonight. So the story I'd like to tell you about tonight, or the information I'd like to give you, really comes from that desire to distill what we know from the scientific literature and make it understandable to people who are concerned, a general public that hears a lot of information from a lot of different sources and doesn't always have the proper scientific level of scrutiny. Lots of people, especially in the area of GMO and transgenic crops, and I tend to stay away from the phrase GMO. It doesn't really work for me, and I'll show you why later. Transgenic crops, a lot of bad information out there coming from activists who really will drive a message of fear and mistrust of a technology that has an outstanding track record. As a scientist, I'm really interested not only in the nuts and bolts of the technology, but certainly its impacts, its possibilities of impacts with health and the environment. And very important to understand those and be in touch with the literature on these issues as well. But overall, I think this is a really good opportunity for us to explore another way to expand what humans have been doing for 20,000 years, and that is genetically modifying crops, changing the genes that are there to make plants that work better for humans, but also that can be used to solve our nutritional issues, maybe address environmental issues. These are all things that we can do using this one extra step of technology. First, I'll tell you a little bit about what I do in my real job, which is actually a pretty cool one. I have a number of different things I do. I have 3 full-time jobs. My first job is chairman of the Horticultural Sciences Department. And I'm in charge of watching the day-to-day activities of 56 different faculty. And we're not a GMO state. It's the state of Florida. We don't grow the corn and soy and cotton. We grow horticultural crops, all which are traditionally bred, all which have no value to companies like the Monsantos, the Dows, not a lot of value to them, at least in growing in our state. We're very much a state that grows crops that are traditionally bred and grows them in various production systems. So my department has a lot of support for farmers in very different contexts. I have my boots in the dirt, soil, at least once a week. We do, our big thing is breeding. So we breed tomatoes, blueberries, strawberries, citrus, tropical fruits. We do a lot of different breeding activities. We're very interested in crop physiology. We have plants that are on the space station. We were the nation's first organic and sustainable undergraduate program. We do plant nutrition. We do lots of cool stuff. So I get to integrate with many different people from many different disciplines and many different areas of plant biology. My program is a little bit different. We work in understanding about how light interacts with biological matter and how light can be used to change the way that plants respond to, say, after they're harvested. Can we make strawberries taste different? Can we change the way they decay with time? This is something that my laboratory does. We're very interested in how we can use natural fruit compounds to retard spoilage, and it works great. we actually have the things that make it taste good have a strong role in retarding spoilage. We also work with this idea of marker-assisted breeding and genomics. We come up with what are the genes that control fruit flavor, and then figure out what those genes are, and then find ways to use molecular markers or specific DNA signatures in different plants to breed them all into one genetic background that makes them taste better. So this is what I do in real life. We use transgenic crops, which is my word that I use for GMO, transgenic crops as one important tool. We use them to understand what a gene does, because to functionally validate so we can have suspicions about what a gene might do, but we have no way to know until we can take it out of a plant or put it into a plant and study how it behaves when you turn it on too much. It allows us to understand what individual genes do. Those are never commercialized, but those are tools that we use in the laboratory. So I have a very strong knowledge of how you make a transgenic plant, how it works, how these plants behave. I've analyzed the fruit quality of tens of thousands of individual fruits and looked for compounds that were made in transgenic backgrounds. So that's what I do. I have a really great group of scientists I work with. And then the other job that I kind of have ended up with, just by accident, is this idea of public science communicator. And about 13 years ago when I was in Madison, 15 years ago when I was in Madison, Wisconsin, I lived down the street from Willie Street Co-op. And Willie Street Co-op was a place that was, as you might guess, very interested in local produce and selling rather refined markets of different products. And I always liked shopping there. The people were nice. It was a really good place. It was convenient. But they had a lot of signs up, anti-GMO, anti-GMO. GMO is killing the world. And I always would ask, well, you guys, I'm a scientist who studies this stuff. I think you got it a little bit wrong. Would it be okay if I came here and gave a talk and we talked about this? And they invited me and we had a great time. I told them everything I know about science, and it was science, 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 and science, science, science. And guess how many people I convinced? 0. And it was because I didn't understand how we would talk to people who were concerned about their food, about the products of transgenic technology and GMO technology. So what I'd like to talk about today is how do these things really work? What are they? What are the real risks and limitations and benefits? Why is there so much pushback against the technology? What are some of the reasons for that? And then I'd like to show you a little bit of a peek into the future about some of the applications of this technology to new kinds of crops and new technologies that have benefits for the environment, for the consumer, for the farmer, and hopefully for the needy. That's the structure of today's talk, is what this technology is. I really want you to understand the nuts and bolts of how this stuff works, so if you're walking out of here, whether you love the stuff or hate the stuff, that you know exactly how it works. Why is there resistance to good technology? Why are there people who aren't so excited about this? And talk about, again, the future of biotech crops. And I always like to show this slide because it really frames this topic for me very well. The irony at the Apple Store. And it contrasts how we feel about technology depending upon the kind of apple that's being genetically improved. If it's the Apple Store, everyone's excited about new technology and new ideas. They can't wait to get their hands on it, and the companies that make it are the best companies in the world. If you contrast this against the new Arctic apple, the transgenic apple that was recently deregulated that doesn't brown, that has an enhanced post-harvest quality, you find we have new and improved products, but nobody wants them and we can't trust them and companies are awful. So why is it that we enjoy technology so much and we trust technology? We use it to enhance our lifestyles in one context, yet in another context find it really objectionable. And there's a lot of reasons we can think about. Maybe we can explore some of those today. And by the way, I neglected to mention, I'll stick around for questions and answers as long as you want to go tonight. I have to be on a plane at 6 a.m. in Des Moines, which means I got to be out of here by 4. So we'll pull that off. So what is plant genetic improvement? And plant genetic improvement I think of as someone who supports breeding programs and other plant genetic opportunities as what are the things that humans have done to plants over the last thousands of years to change the way they behave. And we get more varieties, improved yields, maybe being able to grow in conditions that are challenging, improved nutrition, and maybe even safer products. Safer products came about because humans, as they were domesticating and hunting and gathering the different crops and bringing them into their domestic provision, the crops that made you sick or killed you didn't really move on in the selection chain. Humans have been in charge of selection of crops for, oh, thank you, that's great, for say 10 to 20,000 years now, depending upon the crops that you're talking about. This really contrasts upon what people think genetic improvement is. That this is some sort of Frankenstein process, that this is some kind of horrible aberration that shouldn't really happen. And a lot of that comes from a naturalistic appeal to nature, that nature must rule and that nature has all the answers, when really humans have been changing nature for thousands of years. Hunters and gatherers who were really starting to understand how plants behaved and which plants were good to eat, which ones weren't, which ones you could save and maybe grow the next season. It was that action, that moving away from hunting and gathering to being able to grow plants and farm, that really made civilization explode. Because humans now had time to do other things now that they weren't walking around looking for food, now that they had food and can store food. And so this was a really important watershed in human history, the ability to take crops or plants, wild plants, into our domestication for improvement. So I'd like to ask you a few questions. Let me start out by asking this one. How many people think that this GMO or ag biotech, really great idea? Love this stuff. Okay, quite a few hands. How many people think not such a good idea? How many people think it's a bad idea? Okay, good. We have a couple folks in here. And, and this really does frame very much how a population typically looks. That you got some people who like this stuff, maybe who've really looked at it, others who are much more cautious and wary of it, but the majority of people right in the middle, not quite sure which way to go. And hopefully I can help give you some information on that tonight. What are some examples of some GMO crops? Hold up a hand and then I'll point at you and yell it out. What are some examples? Yeah. Well, Monsanto corn and another company corn, yeah.
Sweet corn by Seminus, who's a company that's under Monsanto.
Okay, well, sweet corn by Seminus. In my state, we don't have any GMO sweet corn, yeah.
Drought-resistant wheat?
No, there's no GMO wheat. Yeah, no GMO wheat commercialized. What else? Yeah. Soybeans, yeah, that's a real big one here, yes. Papayas, Hawaiian papayas, yes. Well, you guys are good. I haven't heard anything goofy yet. Which one?
Sugar beets.
Sugar beets, very good. Well, you guys are really good. Usually I get strawberries, tomatoes, blueberries, watermelons, bananas, and you know, the whole picnic basket full of crops that aren't genetically modified, like at least in that GMO transgene way. Things that certainly humans have had a role in making into hideous malformations from their wild state that we really like. But never really, so we usually get quite a few of those. Well, thank you very much. That's very good. This will be very nice today. The big deal for us, whether we're for or against this technology, is really to remember that we're on the same page. We really are at the end of the day. What do we as scientists and concerned people who are interested in food, what do we really care about? What are our priorities? And I know the things that I worry about the environment. And what is especially living in Florida, which is a very fragile environment, what are the roles and the impacts of farming in that environment? I worry a lot about farm sustainability. How do farmers maintain continual profitable production? And how do they continue to produce the crops that we find important and are very important, not just for us directly to consume, but also in the production of renewable fuels, maybe for feeding cattle. These are other things that are very important. I think that the needy are very important. Every time I sit down and eat something, like tonight, they took me for a beautiful dinner. It was a beautiful salad with a nice chicken thing on there. And I think about the people who may go their entire life and never eat something that good, their entire life. And we have this all the time. And I think that they're really a priority for me in terms of how I want to leave a mark in terms of the future of food. Others, the American consumer, how we can have more choices, maybe healthier choices. Maybe things that we can enjoy that will encourage children to eat better have better eating habits. And I think this is a really important area as well. And this is where I work now for most of my time. So these are our priorities. And I think that if I polled the audience with that one, every hand would go up with every one of those target areas. So whether you're for this technology or against this technology, we all are on the same page at the end of the day. It's just that we have different toolboxes to get there. And I think that that's really my take-home message, is that we have to stop the artificial divisions that are being created by those who are profiting from generating fear around this technology. And we have to start working together to come up with better solutions for our most pressing problems. So let's talk about what this technology is. And some of the basic facts that we can work with on this is that transgenic crop technology, it's really just a precise extension of what we've been doing for a long time. And I'll show you some data for that. The techniques pose no more risk than conventional breeding. And that's a synthesis that comes from the world's greatest scientific minds and medical organizations. That it's an interesting statement because it says it's no more risky than conventional breeding because conventional breeding has risk. That breeders who've bred celery, they've bred potatoes, they've bred wheat, that has had negative effects on human health just by traditional breeding. Transgenic technology, or this adding a gene to the laboratory, is a technology that can have some risk, that you could potentially have a problem, but no more than conventional breeding, because conventional breeding involves mixing things that we have no idea. We'll talk a little bit about that in a bit. In 18 years, we haven't had any case of illness or anything related to this particular this technology. And there really only are several traits that are used in 8 commercial crops. And we'll talk about that in a little bit too. The other one is that these are really the most highly studied plants in human history. That the things that are actually being released are pretty amazingly scrutinized, especially today. It's pretty incredible. So we'll talk about all that. This is a really cool slide to start with. That when we talk about the idea of what is natural, we have to think about where the plants that are the antecedents evolutionarily of those we have today, where did they come from? Where was everything before humans got involved? And lots of people say, well, I only want what's natural. Well, you ain't gonna get it very much in the US of A unless you're eating a lot of sunflowers and maybe some brassicas, maybe some various kind of berries. There's not a lot that comes from the United States and Canada. Certainly we get corn from Mexico. A very small area in Mexico is kind of the seat of corn domestication. Dry bean and squash and other things that came from Mexico. Other crops come from all over the world. And it's really surprising when you see all the different places that our plants had to travel from, our food had to travel from, before it was domesticated by humans, brought into our control, and bred for superior traits that we enjoy. It's really a testament to plant breeders and the people who've done this over many millennia. Right now, we're just doing it with precision. Pretty amazing stuff. We also can see the changes that occurred over time. That corn's great-great-great-grandparent was something called teosinte, an almost useless little stick unless you were able to either soak it and use it, make it into a paste, or as people are realizing now, maybe placing this in the fire and popping the kernels like popcorn. It's a rock-hard little seed case that's almost impossible to eat. But imagine the excitement when the person who was raising teosinte found that one plant that now produced more kernels on that one little stick, or maybe 2 rows of kernels, or maybe found something that didn't have as hard of a shell. You saved those seeds for the next year and grew those. And over the course of 16,000 years, you got to this. And now the hybrids that come from that are really improved. Adding that extra transgene is just one more trait that you can add. So it's a long human domestication process that we're just putting the cherry on the sundae these days. Tomatoes are also a very interesting story that I can tell you about another time. But these little berries that grow on the ground that don't taste so good and have very strong toxic compounds in the leaves have been domesticated through a very interesting route that ended up in our stores today. Many different varieties, all due to human genetic improvement. And bananas? Bananas are the ultimate frankenfood, a whole nother extra set of chromosomes to make them sterile. So there's 1 from mom, 2 from dad that make bananas not produce seeds. So these are all nice examples of genetic modifications, right? They're very different from where they started. So when we talk about, that's why I hate the term GMO. All of these things are genetically modified from their antecedents. That's why I really like transgenic. It's a much more precise use of what we've done.
You have to stop right there because that is the biggest lie that there ever was. When people are objecting to genetically modified food, they're objecting to transgenic plants.
Okay, so we'll come back to
so
that.
that thing that, Genghis Khan, I suppose when he raped
Well,
and pillaged, he was
okay.
engaged in genetic modification too.
This is a great opportunity for us to have a conversation. I'd love to be able to follow up on you. You can be the first person at the end when we talk about it, and I'd love to answer that question. We'll never go through the whole night if we have interruptions. It is a genetic modification. When you we'll come back to that.
Well, that's not the case. When
Okay.
if
Well,
you look at the,
I'll tell you.
if
all right.
look at the patents of these products,
I'll tell you
it
what.
says
I'll tell you what.
specifically
I
genetically
have I'm not going to
modified
argue with you here about that. You can say adding an entire set of chromosomes is not a change in the genetics. I will maintain that it is. We can argue about that all night. I'll be happy to do that, but let's not do it right now. This table really talks about that. How do we, and my voice is cracking, I've been speaking constantly for the last 2 days, so I'm sorry I'm falling apart here. This table is a very interesting table. And it contrasts exactly what we're talking about. Here's what happens when you have hybrids moving through polyploids, which is when you double chromosomes, through mutation breeding, which is where you use chemicals or radiation to damage DNA in a crop to generate genetic variation. When you cross species that wouldn't cross naturally, or this idea of transgenics, where you add a transgene. And now we can talk about some of these other issues. Here you're adding one to three different genes. You know what they are, you know how they work, you know how they integrate into the genome. You know the changes they create and the genes they encode. They're not allowed in organic production, and they're also something that people would like to label. We can contrast that kind of change with a single gene, and it is patentable. We can contrast that kind of change against hybrids, or where we're mixing together tens of thousands to sometimes hundreds of thousands of genes. We don't exactly know what they all do. We know what about 20% of them do with experimental evidence. We can use polyploids, where we can double chromosomes, triple chromosomes. Nobody really minds. We can use mutation breeding with X-rays or chemicals to damage DNA in ways that make a trait that's important, a trait that we like. We don't know about any of the collateral changes that have occurred, but the chemicals or radiation have damaged that one gene that we find useful. That one we don't have much of a clue as to how many things we change, but that's perfectly acceptable. Nobody minds mutation breeding. And this table is really interesting because it sets up a contrast. It's called the Frankenfood paradox. You see this online. What it contrasts is the ways that we have always found breeding and genetic improvement to be very acceptable and conventional, and can contrast that against transgenic, where we're using really what is a much more surgical approach to the problem. And so this has always been very curious to me as someone who works in plant breeding and someone who integrates with plant breeders and others that improve plants. And plant breeders say, I wish I could just move that one trait and not have to bring along everything else. And so that's what we're talking about in terms of contrasting these different techniques. And the plants, as you mentioned, it's mostly sugar beets. Oops, I'm sorry. It's mostly sugar beets, canola, cotton, alfalfa, corn, soy. Those are the main ones. There is some acreage of squash, but very minor, and Hawaiian papaya. And I won't talk too much about the mechanisms of how those work today, but I'll talk about some of the main ones. So how do we add a gene to a plant? And I think this is important to talk about because there's so much mystery around this, and if you look online, they'll tell you you do it like this. And that's not exactly right. Syringes full of scary fluids are something that we usually associate with a yuck factor, which people really do like to associate with this technology. It's actually done in a much more sophisticated way, two ways. What's interesting to note is that every plant that's shown here, none of these are transgenic crops, except for apple was just approved 2 weeks ago. The big deal is how do we exploit this process? Let me do it this way. If we can get the gene into one cell, now you can take that one cell and grow that cell into an entire new plant. And this is an old trick that scientists have known for a long time. It keys off the idea, almost like when you take a cutting and you place that into water and the cells at the bottom of the stem can turn into roots. It's that same idea that plant cells have plasticity. They can take on other identities. And so here you have a plant cell that you can insert, if you could insert a gene into that one single plant cell, you could potentially put that into culture, generate callus, and create whole new plants. And many different varieties of plants have been created by taking steps through tissue culture. So we do this work by adding the gene to that first cell. That first cell has the gene added by something called Agrobacterium tumefaciens. An agrobacterium is a natural soil bacterium that does something pretty cool. It takes part of its genome and sticks it into a plant cell. And once it's in a plant cell, it kind of provides an environment, expresses genes that the bacterium puts there to set up an environment where the bacterium can live very happily. What scientists have done is exploit that same property, and only instead of adding a gene, or instead of delivering the gene the bacterium wants, it adds the gene that we want. And all of the transformation, or all of this that we've done in my lab, is done through agrobacterium, or basically, this is a rather noisy slide, but basically the idea is we understand a mechanism very well by which agrobacterium transfers a gene to a plant cell. Other ways to do this, something called biolistics, or what they call gene gun, where you can take a piece of DNA, project it into a cell, and then sometimes, very rarely, it's integrated into the chromosomes. But that's a whole different thing, not nearly as common. So how do we make a plant from a single cell? And it really falls back to this idea that if you could get it in one single cell, you can make a cell take on an identity of an entire plant. And this is what it looks like in the laboratory, that if you get it into one of these cells, it'll come up as an entire new shoot. So what are some of the main traits? And what we'll talk about are the two main traits that are used in ag crops. They're not really that exciting to talk about, but they're the ones that we always do talk about. And so I would like to give you some ideas as to look under the hood as to how you make something resist an herbicide, or how do you make it resist insects. And these two traits, pretty cool. And this is what you'll see online, if you look online, about corn that makes its own pesticide. And it does. It makes a little protein, that kills a pest. A targeted pest, it kills a specific kind of caterpillar. What's really interesting about this is that it's based upon an idea that we borrow from organic production, which is the use of the protein that's found in BT. BT is one of many natural compounds that work against insects. Plants produce many anti-insect proteins, bacteria produce them, many different organisms produce anti-insect proteins. Plants being stuck in one place have to defend themselves. And they produce these compounds that can be used to deter insect feeding, or even fungus or bacteria. So BT is one good example. It's found in a compound, in a product called dipel, which is a very useful insecticide that can be used on, even in organic context, and organic production on things like cabbage or broccoli, other plants. And it contains this thing called the delta endotoxin, or it's a protein that is toxic to caterpillars, certain caterpillars. The way that it works is pretty cool. So you sprinkle this stuff on the leaf, the caterpillar eats it, and inside the gut of the caterpillar, the protein is processed, which means it cuts off the end of the protein. It binds like a lock and key to a specific receptor, so these molecules in the gut of the caterpillar, and it makes a pore that allows the inside of the gut and the outside of the body cavity to mix. And when that mixes, the caterpillar gets sick, and then it dies. This is how Bacillus thuringiensis works in the BT toxin. What science has done, or what has been done in the laboratory, is engineer this one gene for this protein into the plant. So now the plant synthesizes the protein to protect itself from the insect. So essentially, it's just the plant producing what normally would have to be applied. It's a protein that interferes with the digestion of caterpillars. Benign to non-target organism. It doesn't work very well on other insects, if at all. And it certainly doesn't work on higher animals. And I shouldn't say that, it's very biased. It doesn't work on mammals and birds. BT is a very specific effect on specific insects. The advantages to BT toxin is that when you have the corn make its own protection, you can cut broad spectrum insecticide use. And depending upon the crop, where it is, the type of pest, you see between a 10 and 80% reduction in the use of specific or broad spectrum insecticide treatments. We know that it lowers the cost for farm and for fuel and labor costs. It does have good dividends in the developing world where you don't have other kinds of insect control, and there's no effects on beneficials, whereas the spray plane coming over could. The limitations are really important, is that farmers have to dedicate a certain part to non-GM crops, which takes up their space for something that they may not get as much benefit from. There are pockets of resistance that are occurring. And this is a major problem. When you use this single silver bullet mentality in solving a problem, plants or insects find a way to evolve around it very quickly. So this is where this technology is not working as well as it used to in certain contexts. And it means farmers are spending more time looking for cases where it isn't working and applying old school insecticides. So there are limitations to the technology as well. The other one that we commonly see is this idea of glyphosate resistant, or what we call Roundup Ready products. And I can give you an idea about how these work too. These are kind of very familiar because this is a case where you have soy with corn growing through it, where the corn is not transgenic, whereas this is. So this is herbicide resistant, the corn isn't. And it's been a useful technology for farmers to be able to rid a field of non-transgenic weeds and other stuff. The way it works is reasonably cool. It's a lot simpler and a little more elegant than we might think. This is just a biochemical pathway where A goes to B, goes to C, which makes amino acids and eventually proteins. Proteins that are necessary for life. So if you could interrupt this pathway, you could stop proteins from accumulating and stop this plant from living. Glyphosate works by targeting a specific enzyme that we just call EPSPS for short. It's got a long name. Glyphosate binds EPSPS, and when it binds that enzyme, the plant no longer can convert this product B to product C and blocks that step. And that plant dies. Bacteria have the same pathway, or some bacteria, A to B to C to amino acids to proteins, only their EPSPS enzyme is a little bit different. And their EPSPS enzyme doesn't bind glyphosate. So what if you could take that EPSPS from bacteria and move it up into plants? It's the same enzyme, does the same job, just doesn't bind the herbicide. And that's exactly how this technology works, that the EPSPS from bacteria is put into plants, and now when you apply the herbicide doesn't bind and the plant lives just fine in the presence of that inhibitor. Pretty cool. It's just, and the nice thing about what we've learned from, so the nice part about that kind of technology is it now allows you to create a plant using a bacterial gene that now is resistant to the herbicide. The advantage to that is that you can switch to a much lower toxicity herbicide that's pretty cheap and effective, and it cuts farm costs by saving labor and also fuel. It also really cuts back on tilling and topsoil loss. So there's been some good advantages of the technology. On the downside, weeds become resistant to the herbicide, as we've seen. And if you look at this heat map, what you're looking at in the increasing hotter colors mean more weed species that are being resistant to this particular approach. And the problem is that in some places you just can't even use this kind of technology anymore because the weeds are so bad. Require going back to tilling and other types of herbicides. And that's not necessarily the direction we want to go. But that's what happens when we use these single method silver bullet approaches. So the point is that this is, so that's really how you work with these two major traits. There's BT, which is the addition of this little protein, and EPSPS, or Roundup resistance, where you're adding this alternative form of the enzyme that does the same thing. Almost like changing out your old incandescent light bulb for an LED light bulb. It's just one that works more efficient. The big problem is that this really isn't a scientific debate. We understand how the science works on this pretty well. And when you go through the literature, it really is very clear about the utility, the safety, and efficacy of these products. It turned into a social debate. And so for the next whole part of this, I'd like to examine that a little bit about how that became a social issue. Why is this turning into a debate when scientists feel pretty clean about this, pretty good about this? The scientific consensus is pretty clear. When you look across the world's foremost scientific organizations, in medical organization, they have a very clear consensus that this is no more risky than conventional breeding. So why is there resistance? And there's a lot of people who spend a lot of time and make a lot of money by pushing back against science, by manufacturing risk. There's some people who are on one end who are really working hard to illuminate this technology in a very negative cast, versus scientists, farmers who feel a little bit differently about that. Farmers use it, scientists feel pretty good about the technology, and the difference is that where these folks make a lot of noise, scientists and farmers don't. And the problem is that most people in the middle, like most folks who are just concerned about food, don't know one way or the other. And we end up in this battle of fear against facts. And facts don't win. Facts don't motivate people. Fear motivates people. And so this is where a substantial movement has grabbed a toehold inside this technology to actually turn people away from good technology that can solve problems, the most of it which we haven't even seen yet, by using elements of fear and manufactured risk. And I'll talk a little bit about that. There's actually a very clear movement of a number of people who do this. It's pretty exciting to think about, and we'll talk briefly about how that affects those people in the middle, where 92% of Americans demand to know what's in their food, or this goes back to the DNA labeling issue. That when you can get a number like 92%, it is because many people are being influenced by the fear factor. Oklahoma State looked at this a little more carefully. And in a recent survey, it's hard to see, I'm sorry about that. This bar here says the number of people that oppose or support mandatory labels on genetic engineering. And 82% say that they support labels on genetic engineering. But the folks at Oklahoma State were pretty bright. They asked, who wants their food labeled if it contains DNA? In which 80% said, we need that labeled too. So this really illuminates what this problem is that the people in the middle are influenced by the fear. They don't even know what we're upset about. We just don't want it, which is a wonderful first world perspective to have. So why is there a problem? And this is where it goes back to really what I think of as a scientist as it's a manufactured risk that people are making a profit. It's political motivations and a very strong anti-corporate sentiment, which I understand. I mean, I hate my bank, the people who sell me oil, the people who sell me my cable TV and my cell phone bill. I totally understand that the consolidation of industries has caused problems in the way in our choices as consumers. I get that. But when people the solution isn't to damn the technology that corporations use. Because that's the same technology I'd like to use to solve problems. And I'm not Monsanto. I'm Joe Science Schmo. And that's why this is really a problem for those of us in academic science. A lot of people who are doing this, we can think of some of the folks who are involved in this. Jeffrey Smith down the road here somewhere. Many people who manufacture risk and make a living doing that. Not scientists. Okay, these are folks who are out peddling a message or selling products that have a very strong anti-biotechnology message. Mike Adams even published a website where he said, this is Mike Adams and Vandana Shiva's website. Where'd it go? Their websites where they said any scientists who support biotech should be murdered along with journalists.
Oh, come on.
This is
Who
right
said that?
off their website. I'd be happy to give you the link. Here's her website. Here it is. I'd love to send you the link.
Oh, yeah, I would like to see that.
You got it.
Okay.
All right, so the other big issue on this. And she took it down 3 days after I pointed out that she put it up.
Just for the record, I think about 90% of everything you've said could be proven false.
Okay. And for the record, those of you who support biotech, this is why it's great to have somebody who will interrupt you, because it shows what this movement is about. It's not about science, it's about discrediting scientists. It's not about science, it's about being louder than the scientist is. The facts speak for themselves, and we'll talk about that at the end. I can't wait. So the mom tribe. This is another great example of how our communication has become very insular. And unfortunately, we've been able to be influenced by electronic media to the point where people don't listen to experts. They listen to their yoga teacher, their next door neighbor, their kid's teacher. That the friends and the communities we find online become more influential than the scientists and the facts. And that's one part of the problem. This came from a woman named Heidi from the Center for Food Integrity that said, I'm part of a mom's group. When there's big consensus, I think there's something there. You don't need doctors or scientists confirming it when you have hundreds of moms. Now, that's how you get measles at Disney World, right? You have moms who are making scientific decisions that aren't based on science. They're made upon their feelings and emotions that come from the irrational fears that have been developed by a number of people who've been making money by doing it. And that's how we get into a number of these problems. This is a great example. A lot of you will read about this. People in the anti-GM movement will bring up all the time. Well, there's BT showing up in blood and in fetal cord. And when you look at this paper, it's actually published in a good journal. It looks pretty good until you look into the details. And if you read how they set up the experiment, you find that when we do these kinds of experiments, we first do a test called the standard curve, which allows us to test for the sensitivity of the assay. It tells us when we put in X amount, we can detect X amount. And so they set up this curve, which is depicted by the line here. This comes from their data here. And what they actually claim to detect is down here below their range of detection. That's actually shown here. I'm sorry I cut off that table. But what they're saying is that we've detected Bt in the cord blood, but, and actually what they're doing is reading down in the noise of the assay, which isn't a positive. It's actually saying that there's nothing there. Yet they report that it is there. And it passes peer review, which does make mistakes. The problem is that really what is a non-result gets picked up by the media. And next thing you know, you're seeing websites that are saying detected in children, detected in maternal cord blood, which is a really scary thing to the moms and those who are just concerned about their food. The other one is the famous Seralini rats. Many of you remember this image. And it was shocking when it came out because they said this one got GM corn, this one got GM corn and Roundup, and this one got Roundup. What's missing? The control. So if you do an experiment and use rats that get tumors, if you show the control rat and it's lumpy too, maybe you don't have such exciting data to show, and that's not very scary. When you go back to their data and you look in the tables, you can see that the control rats also get tumors. So why did they elect to not show that one? It's about manufacturing fear. The other really good one, okay, this is showing the same thing here, but this is what you saw in the news the next day. Study shows that this is causing problems, that it causes cancer, that it causes premature death. That's how the media reacted to really what was a non-result. This is another one that came out this week. This one was great. There was a big explosion this week about how glyphosate is a endocrine disruptor. This work came from Dr. Young. I can't remember where she's located, but very good scientist, does excellent work. And I was really excited to see how her data came out and how they were being communicated. What this shows is the growth of cells in culture in different concentrations of glyphosate. And what it shows, or well, Roundup versus glyphosate. So glyphosate is the actual weed killer. Roundup is the weed killer plus the soaps, the surfactants that allow it to penetrate a cell and get in. So what it shows is that you had glyphosate, it doesn't do anything, even at high concentrations that are really high, but Roundup does kill them. And these are placental cells, I neglected to say. In the tissue culture dish, they're placental cells. And so she tested that, and she showed you could kill placental cells with Roundup, but not with glyphosate. And even after time, it was killing it with Roundup, but not with glyphosate. She then was curious, and so what's the conclusion from that slide as a scientist? That it's the detergent in the glyphosate that's killing the cells. You go to the hormone detection, which is the test for the endocrine disruption, and what this shows is that as you increase the levels of glyphosate, the hormone doesn't change, but if you use Roundup, it does. It goes down towards zero. As a scientist, what's the conclusion? Dead cells don't make hormones. No surprise. So what this showed in a very nice set of experiments that went a little deeper than this is that if you use Roundup, which contains a detergent, on sensitive tissue culture cells, there's a threshold where they're no longer viable, which would be expected because that's why we use soaps to wash things. They disrupt membranes. So that was really what she came up with in her conclusions, and very conservative conclusions. But what does the anti-GMO fear factory media say? That glyphosate causes endocrine disruption in human placenta cells, and they show a baby fetus in a womb with a placenta. Looks like human cells are not Roundup ready. It's a gross distortion of what the science says, targeting mothers, targeting those who are really concerned about their food. That really bothers me as a scientist because this is a scientist's very nice work that's being distorted for political motivation. So how do we fix it? There needs to be more communication between scientists and farmers in the community at large. There has to be, these are the people who really understand the science but aren't out there communicating it. And those of you who are involved in science here, it's really important for you to become engaged in the discussion and get involved with discussing the science as it's reflected in the peer-reviewed literature. The companies have never done a very good job in this. The companies that produce these technologies totally dropped the ball with respect to doing good communication before they rolled them out, and did an awful job with that. So how do we fix it? Part of it is better communication. I'll walk you through in our remaining minutes how we can fix this, what are some other technologies that exist that we're not using. And this is the part that drives me nuts when I think about the environment, the farmer, the consumer, and the needy. Places we could be making impacts, but we're not, because the technology is so hard to get approved. This research is done. These are already finished products that are in laboratories. They've already been proven in the literature. So we know that we can grow plants in we know we can do biofortification, meaning adding minerals and vitamins. We know that we can get more efficient use of fertilizers and water. We also know that we can have insect and disease resistance just by adding one single transgene. And I'll show you some examples of that. Golden rice is the example that certainly has its limitations. There's a lot of valid criticisms of this particular product, but it hasn't been pursued with tremendous enthusiasm, in part because of the pushback that has come on it. Really what we're talking about here is rice that now has been supplemented with a gene or genes from plants to make beta-carotene from common precursors. So in other words, this is rice that makes the stuff that's in carrots. And the heat map that's up there in the upper right-hand corner, left-hand corner shows green countries which don't have vitamin A deficiency getting hotter towards those that have severe vitamin A deficiency. It's 250,000 to 500,000 children die or go blind every year, and most of them die within several months. It's been estimated by a study that came out last year that since the availability of this product, that we've lost 1.4 million human life years that may have been saved by vitamin A. And to me, this is an example of how this technology could be properly used. This is to be given away to anybody who is under a certain level of income in the world. And a significant amount of calories come from consumption of rice. The golden bananas are in the news here. This is something that's happening mostly in Central Africa by an initiative from the Gates Foundation. This is an example where a native strain of bananas has had a single gene from bananas added and now produces the vitamin A, now produces beta-carotene that the body converts to vitamin A. This is another one that would work out really well in Central Africa where there's extensive vitamin A deficiency, and actually has been an important subject here at ISU where some of the testing has been slated to occur. Cassava is another nice example, another staple in Africa that also is free from vitamin A and has been engineered in along with iron sequestration and also resistance to a virus which takes a tremendous amount of that crop away from farmers. Allergy-free peanuts are important in the industrialized world because we have kids in every school who have very strong reactions to peanut allergies, and that a GM peanut exists that removes that particular protein that causes the problem. These also are not commercialized, mostly because of the tremendous cost of deregulation. Farmers are really willing to grow them because they would like to produce a safer product. The non-browning potato. 25% of potatoes go to cattle feed because they have bruises that industrialized world consumers find unacceptable. And by inserting a single potato gene in backwards, essentially cancels out the gene that's required to cause the browning reaction. These particular potatoes were recently approved. They also have a gene which limits the production of a carcinogen called acrylamide when you fry potatoes. So you either fry them or chips or fries, you produce a little bit of this unhealthy compound. And this actually removes that particular compound, or it makes a very low level of this compound. So this was an interesting one because it had a benefit not only for farmers, maybe for the environment, because you can produce the same number of potatoes with more potatoes with the same number of inputs that are effectively saleable. The non-browning apple was also recently released. This was last week or two. And this is also another example of a gene that's silenced or turned off by one of the apple's native genes. And that this gene allows the product to last longer and at least be more attractive in a food service application. This one comes from my lab. Strawberries are a very fungicide-intensive crop. It takes a lot of fungicide to grow strawberries. And if you think about the environment in Florida, you're spraying this in very sensitive environments that are close to the water table. And if we could eliminate fungicides, it would be awesome. It would be great for farmers. It would be great for the environment. And we use a plant gene in plants to generate plants that are now resistant to fungal and bacterial pests, which is fantastic. We can never commercialize this. It would just cost too much. But it's good to know in theory that we could do it if we ever got to that point. The BS2 tomato uses a gene from pepper in tomato to allow it to resist different bacterial invasions. So these that don't have the transgene, these do. And this is from its cousin in pepper, tomatoes and peppers being closely related. And this is a nice example because in Florida, conventional and organic tomato farmers use lots of copper. And again, copper in a very sensitive environment would be something that would be nice to get rid of to limit costs as well as limit environmental impacts. Citrus greening. Your glass of orange juice may be an endangered species. We have a disease in Florida that is devastating our fresh citrus industry and juice industry, where trees are infected with a bacterium that's in about 100% of our trees now. Every tree will die from the disease once infected. And so we're looking for solutions, very hard to come from traditional breeding. We do have some good inroads there, and I'm excited about that. But the transgenic trees do really well. For 5 years now, they've lived with infection, meaning they have the infection, but they don't show the symptoms. And how they'll hold up over the long haul remains to be seen. But the earliest they can be deregulated is probably long after the industry is no longer producing oranges. 2019. And even then, we need 60 million trees. So we really should have done this 10 years ago, and there was such tremendous pushback to that. We can produce better oils in soybeans and canola, that the oils have a more healthy compound. So the omega-3s and omega-3s that were previously coming from fish can come from plants, which is really a nice way to have, an acre of plants make as much as 10,000 fish. So it's a great way to integrate healthy oils into our diets. Edible cottonseed can be a tremendous benefit because cotton, you make the cotton, you have to throw away the proteinaceous part of high protein part of the cottonseed, or use it for animal feed. And so this is a major downside too, because that would be great if we could harness that to feed people, but it has a toxic compound called gossypol. And if you could shut down the genes that produce the gossypol compound, now you could have something that humans could eat as a protein source. That one is something that's also in development at Texas A&M. So there's lots more solutions and many places where these technologies could be applied to make a difference. So when we go back to our priorities, when we talk about environment, the farmers, the needy folks, and the industrialized world consumer, those are the priorities of places where these kind of technologies could make a tremendous difference. And it's not necessarily the big ag companies that have to do it if we could allow guys like me to play in that space and the scientists at a place like ISU. These kind of technologies should eventually be adopted once we show that they can work in these contexts very safely and with great efficacy. So the conclusion is that transgenic technology has been demonstrated as safe over many years. Of course, everything has to be considered on a case-by-case basis. The technology has been proven effective in many different contexts, and progress really has been slowed by manufactured risk. The existing products could be very beneficial to the places that we consider priorities. And we need to use all these technologies that are available to solve our world's most pressing problems. That this isn't biotech will save the world. Okay, I don't believe that. But I believe that there will be some important cases where this technology can change the life for some people, some populations that can benefit. Farmers in some countries that can derive some sort of relief. Farmers in Florida that could see their citrus crop restored. It may not change the world, but it can change a world, and if that world is your world, it's an important world. And that's where I get excited for opportunities as a scientist, trying to fix those problems for those groups. And most importantly, it's important for us to be communicating the science, and reading the science and understanding what is the credible science, and communicating that with our friends and relatives. With that, I'll thank you for your time today. It's really important that we're out talking about science and talking about this. And I look forward to answering your questions here tonight. There's some places where you can find additional information. So I'm staying around as long as you are. I'm glad to answer any of your questions. Thank you very much. I'll be glad to come back to you. Let me go to the gentleman in back because I promised him he could be first. Do you have what's your one big ringer right off the top? Like who?
And Bill Joy, who
Oh.
Microsystems, at one point said that genetic engineering, we're talking about taking one gene from another for a commercial purpose, is probably the most dangerous technology since the atomic bomb. Because when these things are released into the environment,
Okay, but what do you have a question? I mean, I'd love to answer questions for other folks. I'd love to answer questions for other folks too. I mean, do you have like one that I could really jump, or would you rather me comment on what you said? Because yeah, you can always find someone who's going to say, yeah, this is the worst thing in the world and the worst thing since sliced bread. And certainly,
Why don't you talk about the the antibiotic resistance genes that were inserted into the various early crops as a way of picking out the cells that you were successful in
Yeah.
getting the genes in.
yes.
And that most of those cells didn't actually produce a corn plant or a soybean plant because their genetics were so messed up. You
Well-
just happened to pick the one that grew like a corn plant.
Well, that's true. All right, so I'll address that. When you use tissue culture to regenerate plants, there can be changes in chromosomes and differences that occur. Occasionally, those will be lethal and you won't generate a new plant. But,
Talking about your gene gun
Okay.
technology where you use a 22 caliber bullet.
No, you don't use a 22 caliber bullet. You use a puff of helium. You create a vacuum inside the chamber. You place your sample. You place particles on top with DNA on them. You produce a puff of helium from a helium tank. Okay. Well. All right. I'm gonna have to stop there because I buy the tank of helium for a gene gun. I'm not gonna argue with you. Okay.
And not just think that somebody that gets up in front of them with a PhD is talking about science when they're really talking about monopolizing technology.
Okay, well, everybody noted. Okay, the guy with the PhD doesn't know what he's talking about. The guy in the audience does. You know, I don't mean to be disrespectful.
Debate you on any stage,
Okay.
any place in the world.
Well, I'll tell you what. Why don't we organize that? And I would happily show up for that. Thank you for coming tonight, though. I do appreciate it. It does help. Well, other questions? You had a question. Sure. Yeah, you have access to the PowerPoint. Absolutely, I'll distribute whatever you want. And I'll be happy to provide the link to the Shiva website. We actually did, when it happened, we couldn't believe it. We made a permanent copy of it and archived it. I'm happy to give that URL as well. It was unbelievable that would happen. Yes?
I'm just curious. I've been reading a lot of articles about the food bank, and it seems like you're always quoted like as I'm
Yeah.
the opposite of whatever she says. So I was just wondering if you could talk a little bit about your.
Oh, no, I'll take care of it. Yeah, so the question was, could you comment on the food babe? Because it seems like she's mentioning me a lot and that we're kind of doing a thing. The food babe is somebody who is an instant celebrity who claims to know about food chemistry and science because she's been anointed as an expert. She has a tremendous, powerful, quite an excellent way of communicating with people. She's really good at that. And she has the food babe army of 5 million people who hang on every word she says. But when she came to my university, she stood up on a stage and lied to an audience for an hour. And I didn't get to ask any questions. And I was really disappointed by that. And mentioned that the next day to the organizers, and they made a big story out of it, and she got a little bit upset about that. Since then, I've been excited to comment on things that she says, especially in the area of GMO, where she really has absolutely no clue. It's nice to be able to assist her with that. But she's somebody who I've reached out to. I don't like adversarial relationships here. I mean, we all want the same thing. I mean, even we want the same thing, I'm sure. She's somebody who could be an excellent voice for science. She just has to learn what that is. So we're working with her. Yes?
I have a few questions, if that's okay.
Well, fire me. We'll come back to the next one. Let me go around, and then I'll keep coming until you're done. I'm happy to keep answering questions.
You are characterizing the anti-GMO folks as anti-science. It was recently published in an article about when Monsanto went to apply for the first round of ready crop back in the 80s. The scientists that wanted to discern its safety said, what about the production of pesticide Roundup-resistant wheat?
Yeah.
And they said, that'll never happen.
Oh, yeah.
Now, would that be called anti-science? Because we all know
Sure.
that things evolve.
If
and
sure.
we know what happens to bacteria when they get
Oh, sure. No, I'd love to answer that. Yeah, we actually, if anybody did say that, they were wrong, and that would be it would be very against what we know in science. We've seen weed resistance to herbicides for the last 100 years, well, the last 80 years, and you never produce an herbicide without something eventually becoming resistant to it. We know that. Glyphosate resistance was predicted, and it happened. It happened later than we thought it would. But it was absolutely predicted 100% by those who knew that we were taking a silver bullet technology and trying to use that. So.
Isn't that what most of GMO science is, a silver bullet technology?
It is. Yeah, no, so the question was, isn't all of GMO technology a silver bullet technology? The reason it's a silver bullet technology is because to make it bigger than that requires more deregulation and more time. And to deregulate one trait at a time was the only way these companies did it. Because of the need to put a product out that in a regulatory system that was almost impossible to navigate. And I think if we had things a little more scientific and reality and realistic, those things would have had multiple traits and stacked traits in the beginning. I'm with you. The anti-science comment, I do apologize for that because I don't like to throw that term around. It does happen. I see, creationist billboards. I see people saying, don't vaccinate your kids. I see people saying the earth is getting cooler and glaciers aren't melting. And I call that anti-science too. I do have to be a little more careful about that term because it is divisive. So I do apologize for that. But
Don't put all me in the category of climate science deniers, please.
I don't know you though.
Yeah, well, so I think it's a little different.
Well, and we can have the conversation.
What you do when you make these analogies that are sort of very non-scientific. An analogy is a linguistic structure that creates an equality where there is none.
Well, if we look at the consensus of science, well, we look at the scientific consensus and we look at the peer-reviewed literature, which is what I have to operate from as a scientist. Presenting the strengths and limitations. I mean, I don't just advocate for the technology, and I'm happy to tell you the downsides. There absolutely is 100%. There's very strong consensus that's occurred around those areas, and I think that, and certainly there's room to think about them.
Monsanto did deny that would happen because they
Well, we should follow up on that though, okay? Because
send you that link.
you can send. Well, I can send you the scientific paper, and we can. I'm not so excited about a link from Jeffrey Smith, but I'll be happy to show you the scientific reports that showed that people going back even into when these things were under development always predict when any herbicide is overused, you have that kind of result. I'll come back to other questions. Any other questions or thoughts? Oh, yeah, thanks.
I don't know. I mean, you didn't really get a chance to explain the whole shooting helium into it, so I was just a little more interested in what that actually was.
Okay, so she said, what's the deal with shooting helium? So to answer the gentleman's comment in a little more resolution, the original gene gun used a gunpowder charge to create a power, some sort of power that would propel projectiles into materials. That was the original design. But for the safety of those using it more than anything else, there was a tendency to get away from gunpowder and hook this thing up to a very high pressure directed helium surge in a vacuum. And so you would hook up your helium tank, you would have your sample in a chamber, and it would sit below where the particles were on something called a rupture disk, that when you charged the system and pressed the button, it would go pop and your sample would jump because it just got shot with helium in a stream of particles that went into the tissue. So it was a helium-powered projection, not necessarily a 22 cartridge. Maybe in the original constructs that was how they did it, but that isn't how it's done at all in the companies afterwards. Yeah, thank you. Now that wouldn't wash in most places. OSHA rules. Yeah. Hi.
How does our body digest GMO product compared to non-GMO product? So you say it can spoil, and then compared to one that's
I'm so glad you asked that. So the question was, how does the body deal with a GMO product? And so what's different in a GMO product? Let's talk about, like, say, BT. People worry about that. It's an insecticide, right? It has DNA that's different, and makes a protein. And those are digested just like any other DNA and proteins. They're digested just like any other nutrition. There's nothing that discerns that particular piece of DNA from anything else. And if you think about it, we don't eat sterile food. We're eating bacteria and fungi and all kinds of other goodies in every bite all the time. And so we're constantly taking in bacterial and foreign DNA and foreign proteins, and they they're digested like food.
But what if the fact that it capers down on its own or it takes longer, then how does our body digest that?
Well, I think so the question was, what's about the fact that it doesn't break down as well? And you're referring maybe to some of the future crops that can last longer because of and that's, to me, that's an exciting thing because we lose 40% of food to waste and spoilage. And this is a real problem in the developing world. And so could we make things longer with genes that break the senescence process inside a fruit or vegetable to make it last longer? But that's a cellular process. What that means is that you're changing a gene that affects a protein that has some sort of role maybe in regulating the breakdown of those cells in the cell death process that's very naturally orchestrated. And so it really is just affecting a cellular process. As soon as you eat it, you break it down like anything else. And all that has to be tested before those things are approved, even though there's no plausible reason why it wouldn't break down. Any other thoughts, questions? Oh yeah, way in back.
First, thank you very much for your presentation. I thought it was very clear, so thank you.
Oh, thank you.
I was curious when you developed GMO, so if you add a gene to make corn producing a certain compound for protection, have you ever noticed any trade-off? Like, does the plant have to, like, instead of putting out a lot of energy in producing these compounds?
No, it's a great question.
Trade-off in place of like physiology.
Yeah, so the question was, do you see a trade-off physiologically? Oh, first thing she said was, nice, clear talks. Thank you. I had to do that. The other thing she asked is, do you see a trade-off that when you, say, create an anti-insect protein or maybe a herbicide resistance protein, do you see something else change? And the companies that look at these things look very carefully, especially today, that every plant that comes through these processes is the whole genome is sequenced, you know exactly where these things integrate, you know lots about it. You also take a very careful look at yield compared to what they call the isogenic line. So you have a hybrid that performs very well, and now you add a gene to it, and you can test side by side whether they're the same. You can't have a yield hit because of your trait, unless of course maybe it increases, you know, water use efficiency or something. Actually, that's a good example because farmers see a little bit of a hit in the productivity of those lines, but they do see better efficiency of water use. So in times of drought, it's a great score, but not always but no real yield difference under non-challenging conditions. That's a really important thing to keep in mind because especially as we look at more metabolic engineering, meaning we're changing things associated with metabolism rather than enzymes that are affecting other traits. Maybe endpoints like the beta-carotene and other, we may expect more of a metabolic hit, but we don't see that too often. I never see it in strawberries, and we make lots of them. So, yes, I can.
You kind of give the audience a feel for how many plants would be tested conventionally to arrive at a product versus how many transgenic constructs might be tested to arrive at a plant that could even start the evaluation of product evaluation.
Yeah, so to think about this, you're thinking about in the context of when you do traditional breeding versus, yeah, so traditional breeding is really awesome, and I love plant breeders. I wish I was a plant breeder. And those of you who are still in school and thinking about a career path, please learn plant breeding and be one. Not only do we need great young plant breeders who have a great eye and concerns for different issues, it also is a very lucrative business these days in terms of, you know, you make a great living doing it. Companies love it, and you have a great job in universities, lots there for you. Anyway, so plant breeders, the whole trick is to take two plants that maybe some genes that are favorable and some genes that, so two plants, one of them has good disease resistance, another one's got great flavor. But this one's got bad flavor, this one's sensitive to disease. You might mate those two by, you know, doing plant sex, and then take those seeds and plant those. You may have to screen 5,000, maybe 10,000, maybe 100,000 plants before you find one where those same traits are in one genetic background. And that's really the big battle here. Maybe not so bad if you're breeding something like tomatoes and you can generate a generation every few months. But what if you're growing citrus trees? Getting the next generation of fruit takes a long time. So when you compare that and our efforts to get rid of eradicated disease using traditional breeding versus adding a gene, which might take a year or two to get that thing in and get the plant going, it's tremendously more resource sensitive to be able to use a transgenic approach. Can you imagine how much it costs to grow 10,000 citrus trees? In terms of land, in terms of fertilizer, in terms of human input, diesel, it's hundreds of thousands of dollars, and it's millions of dollars to make a new citrus tree. Making a new strawberry might be 500,000. It's very expensive. Yes.
How would you reply to someone who said, "I'm morally opposed to moving genes from one species into another, from bacteria to plants. I don't care if any food is derived from that process"?
Yeah, and then, so this is a really important one. It's, how if I'm morally opposed into crossing species, but you're even talking like jumping kingdoms, moving genes around. And I think that the best example is that many of the things that we enjoy every day are combinations between species that maybe wouldn't combine on their own. The strawberry is a great example. The strawberry that we enjoy every day came about because of colonists bringing strawberries to Europe in 1500s and a spy who went to Chile in 1714 and brought back strawberries. They both ended up in the French botanical gardens, and these two different species were able to combine to make this hybrid. So they were sexually compatible, so it didn't seem so weird, but it was an example where you could cross a species barrier. But people who have other objections maybe to the more refined ability of moving something by recombinant DNA technology, I would say nothing more than let's look to medicine. Here you have the ability, we used to have problems with diabetes, where you would have to go to a slaughterhouse and collect pancreases from cows to generate enough insulin to treat people. With our insulin problem, with our diabetic problem today, we'd never be able to do it. There's not enough cow pancreases around. But by taking the gene from humans and putting it into either bacteria or yeast and being able to grow those microbes in gigantic fermenters, you can generate monsters of life-allowing insulin, monster amounts of insulin that now can be used to treat a human condition. So here's a great example. By taking something from one species and putting it into another can serve an entirely different species. So there's a lot of good examples of how this can have tremendous benefits. Any other thoughts, questions? Wow, might get out of here early, yes.
The speaker that was here and presented her viewpoint at the meeting earlier this month
Yeah.
had a theory that the GMO modified vegetables, fruits, and grains all had less nutritional value.
Yeah.
I don't know where she come up with that.
No, that's Dr. Yeah. Yeah, so the question was, or the comment was, Dr. Shiva had described that plants that come through GMO technology have lower nutrition value, and on a case-by-case basis, you can show that the transgenic crop typically has the same levels of nutrients as their isogenic equivalent, as we say. The one without the transgene, the one with, make an equivalent product. And that's almost invariably across the board. There's not any real substantial difference that you can find. Now, you can find places online that will tell you that. One of them was very radical, and I actually challenged them. I said, tell me about the plants that you grew. Let's do this test together. And we'll use your laboratory, we'll use my laboratory. And we'll come up with a good final answer for everybody. And they said, absolutely, we'll do it. And I said, okay, put me in touch with what the seed corn types are, and we'll grow some of them in fields that I pick, we'll grow them in fields you pick, we'll use your lab and my lab, and I'm paying for all of it personally. Not University of Florida, not anybody, me. And we got to talking about that, and then they sent me a letter that said, well, we really don't want to do this. So when you hold their feet to the fire, they back out. And there's other fun cases of that too. Yes?
I think she was referring to an article from the British Journal of Nutrition that was a review study of, I don't know, from 60 or 100, right in there. I mean, it was definitely double digits, several dozen of studies on organic versus non-organic. So she contemplated the non-organic and the GMO, but that's somewhat reasonable to do. I mean, it was remarkably higher levels of antioxidants in the organic.
Yeah, that's true.
Peer-reviewed journal, it was
Absolutely.
an article of dozens of other articles.
Yeah, it's what they call a meta-analysis.
Yeah. Pretty impressive.
Well, it was a meta-analysis that looks at, it takes a whole bunch of studies, kind of puts them through a funnel, and then comes out with what the outcomes are. And they showed that in specific crops, I can't remember which ones they were, you did have higher levels of antioxidants associated with organic production. Yeah, no question. The numbers weren't giant. They were 20, 25% in some cases, which you expect. I think that's totally expectable. But you typically do increase your amount of antioxidants under stress conditions. So if you have maybe more insect pressure or other issues like that too, which I'm a big fan of anything that does lead us towards fewer farm inputs. So I support our organic farmers big time. Big time.
Can I get my next?
Sure.
I divided it back. Yeah, you mentioned that it'd be really great if we could have dialogue, and there is a dialogue
Yeah.
going on over in Memorial Hall. There's a group.
Yes.
I just visited there. I sent my reporter over there because I couldn't be in two places at once. Your talk was scheduled conveniently right at the same time as that.
I know.
And
You can thank them for that.
I don't think so. And
No?
it's been planned a long time, I think.
Oh.
The
Meh.
the Department of Ag Sciences had agreed to participate, here at ISU, and agreed to participate in this forum to talk about the testing of the transgenic bananas. And then they suddenly withdrew. They didn't want to do it. Then after she was talked, they said, okay, we will do it. And then they withdrew. And it seems to me that the ISU College of Ag and Life Sciences is just nervous about sharing conversation about these issues with people who are not concerned about biotechnology. Not only we're talking about all of the physiological consequences of biotechnology, but there are so many others. There's environmental
Sure.
consequences, there's economic
Yeah. Sure.
consequences, and all of these things are giant ethical issues. And the College of Ag and Life Sciences, turned its tail and ran it
Well,
as far as I can see.
Can I tell you? I'll tell you what I know about that. Jen, who's involved in the Sustainability Ag Club here, she emailed me maybe 2 weeks ago and asked me if I could participate with them. And I'll head over after this, yeah. And she, I'm excited to participate. She asked me to do it, glad to do it. This has been scheduled since last year, so I have no idea. We didn't plan anything for tonight for sure. It's really kind of fortunate. Well, yeah, they're right. But to address your point, I think they were excited to participate because really what they were asked to do here at ISU was to test the bioavailability of the beta carotene and some of its derivatives. And the reason they chose ISU to do it here is because there's a scientist here who is a world expert at being able to do that. And she.
One of the issues that they have is that the bioavailability of beta carotene depends really highly on the fat content of one's diet, and the fat content of the American Iowan diet is way different from the fat content of the Ugandan person's diet.
Well, but that's taking it in a different direction. The question was, so if we take this as scientists, we have to look at this in bites. We have to say first, regardless of other factors, if we administer this, what is the bioavailability? What if you test it in people who have fatty rich diets and it turns out they don't absorb any of it? Then you're done right there. You don't even have to worry about the other questions. What it was is a world expert is here and they asked her to do those experiments, and she's an expert at that detection. And when that's what the job was from the Gates Foundation effort to put these bananas in Uganda, they have to do some tests like this. And she's not a person who is interested in debating GMO or the economic consequences. Her job as a scientist is to do her best job to examine the bioavailability of those compounds. Now, if she turned down that opportunity to do it, or if Gates Foundation didn't hire her to do that, people would be saying, you're not testing it enough. You're damned if you do, you're damned if you don't. So you might as well do and be damned. I mean, I give her a lot of credit for at least taking it on. Drop her an email, and she'd be happy to answer that. I think the problem. Well, because she's a scientist who's here with a job to measure chemicals in the blood. She's not somebody who's prepared or has a background to be at the front ends of a public controversy. And I understand where she's coming from, And you know what? And that's probably, and that's. I think now, if I was the scientist, I'd take this on head on, because this would be an opportunity for me to engage in an audience in talking about how these things are tested, the things we're going to look for, the things we're not going to look for, why it's important to the entire study. I think I would have taken that head on. But I'm always really comfortable with that. Scientists are weird people, and we've been selecting for a group of people who aren't necessarily excited with interacting at the public interface for a long time. Can't expect people who are excellent in the laboratory to automatically feel very comfortable engaging in a public conversation with a topic that they may not know the entire history.
Do you think there are ethical issues surrounding biotechnology that have to be addressed?
Well, we have to be more specific. Because I think the ethical, for me, it's a little bit different, and I'll flip that upside down, unfortunately. For me, the ethical one is, should we let a vocal minority stop good technology from reaching the world's hungry? And should we let a vocal minority of non-scientists steer research? I mean, those are my ethical questions. To me, the ethics are crystal clear. We need to be using the absolute best tools to solve the world's problems, and this is one of them. And so, to me, it doesn't...
Yeah, I mean, you know, Santa Shiva has a very different approach to that ethical problem. Do you think that ethical approach is valid?
I don't necessarily agree with Shiva on these things, because she hates corporations more than she loves people. Her whole thing is framed in such an anti-corporate vein that she doesn't recognize the human suffering that piles up when we fail to act with our best technologies. We can solve problems in the world safely and effectively. We can do it. Guys like me want to do that. I'm not Monsanto. I'm, Joe Science Schmo,
I know you guys are all very,
And that's where our hearts are.
save the world, but maybe
Well,
more people save the
I don't want to save the. I mean, I'd love to save the world, but I'll take changing a western province of Zaire just a little bit for a family. I mean, I don't need much. I'm in this for the right reasons. And so when we talk about ethics, you got to understand how frustrating it is for a scientist who has a toolbox to not be able to use it. What if we, through conventional breeding, a breeder mixes a kind of rice with another kind of rice that have never been crossed, and the rice comes out with beta carotene, and it has the beta carotene in it, and he says, "Eureka! We can feed this to all the people in Asia who eat rice who are suffering from vitamin A deficiency."
Monsanto comes in and says, we will encode that genetic sequence and make it ours,
And-
and we'll have to pay for it.
And how would you feel about that?
I would feel really lousy.
I would be outraged. Are you kidding me? If a company came in and said, "We're going to stop you from using that good technology,"
That's what happened to Santa Shiva. That's why she's anti-corporate, because they did that with basmati rice. Maybe it wasn't Monsanto, it was a different corporation.
well,
They did that to basmati rice. The Indian people had bread, and it was a traditional, beautiful rice product, and they came in and they were going to genetically encode it and then patent it and then make it theirs, and they would have to pay for it.
well, there's a place where reality separates from the fiction on that particular statement. We can talk about that. There's nobody who's if you create a genetic change. All of this stuff. If you'd like, you should go visit the seed repository over here. There are massive, beautiful seed repositories throughout the world for public domain crops. We can get stuff that we don't have to pay licensing fees for. Special changes that are made in crops, even the stuff that comes out of University of Florida, like strawberries or blueberries, that's licensed, patented technology, not GMO. But it's because it costs $1 million to make that new blueberry variety. Should we have to give it away? And so there's a lot of unusual nuances here. I'd love to follow up with you on all this stuff. You seem like a wonderful, nice person who just has questions, and I'm glad to talk to you. Absolutely. Any other thoughts or questions, ideas? Yeah, I hear you. Well, and everybody is welcome to stick around.
You mentioned that the BT crops have no effect on beneficials, and I really want to know where, in fact, in research, is the basis of that statement.
Oh, lots of. So the question is that BT, how do you know it doesn't have an effect on beneficials? Excuse me, my voice is totally shot. There's a lot of ways that that's known. From either observations in the field, but also just from direct feeding experiments. You can feed certain animals or insects nothing but BT pollen, and at times when you get super high doses, they might show unusual behaviors, but they're at doses that are thousands of times higher than they actually encounter in the field. What you see in the field around BT crops is an expansion in the biodiversity of other beneficials that typically aren't there when you come over with a spray plane. And so it's, or use other kinds of pest control methods. So these have actually been really well tested, and there's a lot of nice ecological data from BT. So I'd love to show you. Yeah.
I was just
Yeah. Sure. So thank you so much. I'll pay you later. So the question was, could you touch a little more on what's happening with oranges? In probably around the year 2000, a tree was brought illegally into the state of Florida and planted in a Miami backyard. And on that tree was bacteria and an insect that could transmit it. And that bacterium has now been transmitted to almost 100% of Florida citrus trees. To put that into context for you, if you drove from here to Chicago, you basically would be driving through an equivalent space of our citrus industry. Citrus on both sides of that road, beautiful dark green trees with beautiful oranges, beautiful blue sky. This is what I moved into when I moved to Florida. Now when you take that same trip, it's gray skeletons of old sick trees with sick tiny little fruit that are green. The disease is called Huangbang Ling. It means yellow dragon in Chinese. We call it HLB, or citrus greening. And it's a disease where a bacterium is moved from tree to tree by this insect, and their insects are everywhere. And it infects the tree gets very sick for a long time, may have the bacterium for 5-6 years and no symptoms, and then suddenly collapses. So this is a really important disease, and the problem that we're having in Florida is that it isn't just a farmer going out and picking a basket of citrus, throwing them on a truck, and calling it their crop. There's a huge infrastructure of packing, of juicing that is supported by this. And the problem is that you can't keep a juice plant open if you're not bringing in X amount of oranges. So our deficiency, we need 1 million boxes of citrus to stay productive, stay in business. This year, we had 1.2 million. So next year may be the last year for citrus out of Florida. It's devastating. And when you look at the families that are the 5th generation farmers that have had wonderful operations, who are awesome in their communities and do a beautiful job producing healthy fruits, it's heartbreak what's going on down there. And I'm proud to be on that front line. We have transgenic trees that have come from Texas A&M that are 5 years in the ground now, have no symptoms. And we have 3 different genes from University of Florida that look fantastic, that the trees they have the disease, but they don't develop the symptoms. So we think that the transgene plus enhanced nutrition plus better genetics in the first place, plus managing the psyllid, the insect in different ways, plus maybe some antibacterial treatments together give us a solution. And it's really an important point for this whole discussion, that there is no silver bullet, that transgenics and the ability to make a GMO tree or a GMO plant is just one way in which we're going to help to solve problems, and hopefully in the citrus industry. That might be the first one that really gets public approval. Yes?
So my grandpa lives in Florida, and he's really worried about this because he loves his orange juice. And he tells me that they're ripping up groves and planting new trees.
Yes.
And I'm filling them in on as much as I know. And so I'm wondering, how long do you think that will prolong this issue? I mean, do you think we can reach 2019 without, the whole industry collapsing, basically?
Yeah, it's up. So the question is that there are some approaches that are being used in Florida to combat this, and part of it is just planting new trees. And farmers used to plant a tree and let it grow for 30 years and harvest every year. What we're doing now, and what the strategy is to plant trees on smaller intervals, grow them for 10 years, tear them out, and put in new ones. That's the thinking. And to use really intensive psyllid control and really intensive fertilization regimens to help them grow through it. Maybe not the best thing for the environment, but that's currently how that's going to work. We do have traditionally bred trees that look fantastic, I'm hopeful, and also some rootstocks. So we can take a root and graft on a new scion, and the root does impart some resistance. We're really crossing our fingers on lots of these things. So it's a really interesting time to be involved in Florida citrus. The organic guys really took it the hardest because they really did a great job of managing their grows, doing the best they could in nutrition, and my hat's off to them. They did a beautiful job. Yes?
You said you're using bacterium to create your transgenics. Why did you decide to use those instead of engineering things like talents or CRISPRs?
Good point. So you've opened the next generation of what we're doing in biotech. So the future of biotech is a little bit different in that there are gene-targeted technologies that will allow you to essentially edit a genome. So if you can think of all the genes in an organism, maybe printed out on paper, would stretch for about 18 miles in a standard 12-pitch font. one side of the page. That we can go on to page 498,492 and change a few letters in a sentence. That's how powerful this new technology is. And that's coming. And actually it's here now in many different places. We use the transgenic approach because it's very easy and agile for us to do it. And we don't have these other technologies validated and working well in our context. So, but that's the next generation. Yes, way back.
Would you mind commenting on two things?
Sure.
First of all, the WHO, World Health Organization, declared this week that glyphosate is a carcinogen. And then we have the National Soil Tilt Lab on campus here. And 2015 was declared a year of the soil. There's patent number 7771736, which Monsanto was granted, and it declares or lists the bacteria species that Roundup kills in the soil. Those are quite a few of the very important species, such as trichoderma and pseudomonas. So, comment on this.
Yeah, so the first one goes back to this week. There was a release by the, and I always forget the acronym of the organization, but it's an organization that provides guidance on the way that they list the relative toxicity of different chemicals and frames it in the context of carcinogenicity. And they reclassified glyphosate. So glyphosate has been examined by many different agencies over the years. On its MSDS and other places, it'll say either non-carcinogenic, or they have very good examples of when it becomes toxic. We know this stuff pretty well. They reclassified this not as a carcinogen, but as a probable carcinogen, level 2A probable carcinogen, which put it on the same level as aloe vera extract and one below sunlight. So it says that there's a potential to do that. They did that without seeing any new data. And so I was really curious about how they arrived at that conclusion when all the data previously have supported a different interpretation. But they are coming out with the full report, and I'm looking forward to seeing why they decided to change that. The other question was about about this being the year of the soil, and that glyphosate having effects on microbial communities and microbial composition of soil. Right? Is that what you're going for? And I don't know that exact document, and I would have to look that up to take a look at it, but certainly something like glyphosate, which it's a compound, it's an amino acid ortholog, could have effects in bacteria because they possess that same kind of metabolic pathway that's in plants. So you might expect that there would be some sort of microbial effects.
It also takes out the lactobacillus and Helicobacter pylori, which we have in our gut, and they're very important organisms.
Those are important organisms, but the big problem is, the big question I always have about the questions of glyphosate on gut bacteria is, how do you consume glyphosate? And there's very little of it used on a field. The amount of glyphosate used that's sprayed on an area that might give rise to a kilogram of soybeans is minuscule, almost non-existent. And for that, and that's sprayed weeks before the beans are on the plant. So there's very little likelihood, and it is tested. They do spot check for glyphosate on crops with certain specific limits. And you do detect it occasionally, but it's always detected at levels that are pretty low. I mean, I think when we looked for it, you could find 1 or 2 ppm. 20 ppm is the limit, and you do see that occasionally, but it's only when people use it, I think, are using it incorrectly or spraying too late. I think if used by label or by direction, it's difficult to get any level that would be that high. And to have levels in the body that would actually be biologically relevant, you'd have to, I did the calculation, you would have to eat thousands of kilograms of soybeans to achieve a level that was biologically relevant. But it's an interesting point. Yes?
Do the big seed companies seem to be making a better effort to reach out to the public? I saw this documentary recently of GMOs, and they tried to talk to Monsanto and Syngenta, Pioneer, and nobody would talk to them.
Yeah.
And I kind of believe that nobody would talk to them, because people are so touchy about what messages are.
Yeah, I think so. The comment was the company's actually doing anything to maybe reestablish a public face. I think they really are. There's people that are now being hired for millennial engagement and for social media engagement and many different areas that they should have done years ago. A lot of the documentaries, I mean, I think if someone showed up at the front desk of the university with a camera, they would say, get out of here too. There's certain protocols you have to go through with getting permission, and you know. So it's one of those things. I think for documentaries, it is very nice to have someone come up in a security outfit and say, nothing, no filming here, you know, just to add to. But who knows? I think that if you talk to Rob Fraley today, the guy who invented the first GM plant, who's still a higher up at that company, he'd come out and be glad to talk to you. So I think the companies realize that by being difficult and by hiding from questions, they created more suspicion and more problems. And that level of transparency is very important. Yes?
In your slides, you said somewhere around like 92% wanted labeling for GMO.
Yes.
Whether they like GMOs or not. Why do you feel that labeling or mandatory labeling on GMO products hasn't become standard?
Yeah, I think it's because, so the question is, why hasn't mandatory labeling become standardized? It hasn't been common practice. I like to think that the main reason is because it's not a scientific evaluation, that there's already a very rigorous system by the FDA that's in place that says if something has a compound that can adversely affect biology, that you must include it on the label. That's already in place, and that's why something that's been, if you have lunch meat that was processed on a mechanism that was used on peanuts or something that may have contained wheat, you need to have that on the label. So the label instructions are already really clear. If there's some sort of risk, you have to divulge that. And there hasn't been any solid risk that's shown from a BT protein or the gene that encodes it, or EPSPS. So that's the one major reason. The other thing that's really been nice has been the reaction of the market to produce either non-GMO project products or organic products, where those sales are going up like crazy. Because people who are not interested in transgenic products that come from a transgenic plant have other choices, and they're clearly labeled. So, and that's just kind of my view as a scientist. I think as a person, I'm very uncomfortable with having my state government set up another agency to label and enforce and test and do all that stuff for something that doesn't have a health risk. So that's kind of why I'm a little bit leery of that. And I'm not a big fan of any of the remedies that have been proposed. The other big point about the labeling initiatives is that all of them have had awful language. And I think if they were written by scientists rather than lawyers and activists, they probably would have went through. It's just all the exemptions, all of the non-transgenic crops that would have been included. Hawaii just banned seedless watermelons because they were made by chromosome doubling, which is forbidden under Maui's new rules. They don't know that yet. So there's a lot of nuance in those regulations. That one of our citrus plants that may resist greening is what they call a somatic hybrid. They actually fuse the cells of two different citrus types that you can't cross sexually, fuse them together to make something that you could then regenerate into a new tree. And now it has chromosomes from two different kinds of citrus, and it seems to be resisting the disease. That would be called a GMO, even though there was no recombinant DNA used. So, any other questions, thoughts? Yes?
So, as it sounds like you do support the organic industry somewhat, but also are a scientist interested in the use of transgenics technology, how do you reconcile supporting the organic industry, which is by USDA definition?
Yeah, so the question is, I seem to at least discuss that I support organic farming and organic production. I also really am interested in transgenics. How can you reconcile that? And I think it's really easy to reconcile scientifically. That I want the two best methods that have the lowest impacts on the environment. So give me a plant that grows with less nitrogen requirement, maybe less water requirement, maybe makes its own protection from insects and fungus, and put that in an environment that you can grow productively without a lot of synthetic inputs, costly inputs. That's not too bad. I think it makes 100% sense. I also think that both of these things stand alone pretty well too, that anything you can do to create plants that help farmers remain productive, I'm all for it. Yeah.
So just as a follow-up question, do you think there should be a new labeling for transgenic but also organically produced?
So should you make a label for something that's a transgenic and organic production? I like to call it morganic. You actually can get one better. It's because I think that this is the marriage that we'll see in 50 years. When your grandkids watch this video, and they go, look at when he was in college, they'll say, hey, there was that guy on stage who called that first. I really do think that these are very compatible technologies and ultimately will be the synthesis of our efforts. Yes?
I simply have to disagree vehemently with your characterization there. Because organic farming is entirely about biodiversity, whereas Roundup Ready corn and Roundup Ready soybeans have created a monoculture across our entire state and beyond, where there is no biodiversity. I mean, the two couldn't be more different on the basis of biodiversity, which is the root of all life.
Well, all right, so but see, you missed my projection there. I'm not talking about let's stick, because you couldn't grow
And that includes the little bacteria in the soil.
but you couldn't grow a jillion acres of a transgenic crop in an organic system very easily. I think an organic production system that, let's say my strawberries that don't require fungicides and don't require any kind of pesticide or any fungicides or antimicrobials, those would be tremendously valuable to an organic farmer. The tomato that uses a pepper gene so that you don't have to use copper or any kinds of antimicrobial treatments, those would be wonderful for organic farmers. So they're transgenic, yeah, they're not making a monoculture, they're giving you the ability to use more diversity in your particular operation. I mean, that may be a really nice way to think about that. Let me go way in back, I'll come back to you, Candy. Yeah.
Well, I kind of in regards to that, though, don't you think when Chief said that when Roundup, when genetically engineered crops came out, we turned into a monoculture? So, say in 1990, this whole state was covered with oats and alfalfa, wheat, corn. It wasn't. We were already.
Yeah, the hybrids were already very.
Synthetic chemicals. I mean, I don't think you can blame transgenic crops on our current crop.
No, I'm with you. I think that what you had here in the state was, or what we had anywhere, were hybrids that still were from a very relatively narrow germplasm. Your Candy could probably address that.
I'd like to follow up just where I was headed. We had a monoculture too long before we had GMOs. We lost wheat and small grains when we went to soybeans. They weren't GMO then. We lost them because it was a choice in cropping systems and convenience for production. It had nothing to do with GMOs.
Okay. No, very good. Thank you. Wow, we're doing all right. Wow. Yeah. Yes.
You talked about transgenic apples earlier.
Yes.
What are they trying to do with those?
So the transgenic apple was made by Okanagan Specialty Fruits. They're a company of 4 full-time employees. What they did was put in a gene that is responsible for the browning reaction. So the interaction between compounds in the fruit and air that would lead to browning. And what they did was knock out that enzyme or suppress that enzyme level. So these apples, you cut them, you leave them out, they never turn brown. It's the same exact enzyme that was turned off surreptitiously, is that the right word? But by accident. A breeder in grapes in California was walking through his vineyard in 1961 through what were purple grapes and noticed there was one part that was hanging off that was variegated and unusual. And when he cut that off and stuck it on a rootstock, it produced yellow grapes. And they used these to make golden raisins. When you look at this now with molecular tools, it turns out it's the same gene that had a naturally occurring mutation that turned off the polyphenol oxidase enzyme that is the one responsible for that browning reaction. The advantage with apples is it makes them appear reasonable longer, so edible longer. So especially in food service applications and maybe in cutting it up and taking it to lunch. Like I eat apples constantly. I'd love to have one.
But does that mean that they're actually still good? Because isn't browning like a good indication of when the food goes bad?
Yeah.
Like is that kind of a negative?
No, well, you would have a problem discerning it strictly from the browning, but you would see water-soaked lesions and other indicators of damage. Certainly, if it was no good, you'd be able to tell. I've actually eaten this thing. I've eaten the Roundup, not the Roundup Ready, the Arctic apple, and it tastes like a regular apple. They tell me it tastes better, and it tastes like an apple. Any other thoughts, questions? Wow, can't believe it's really cool. Yes.
So less than 2% of
And this is, so the question is, when you have people who are very popular, who have tremendous followings, who are talking about information that isn't necessarily consistent with the science, and scientists and farmers who are using products and standing for science that is reflected in the peer-reviewed literature, how is someone like you play a role in shifting that needle, that as people are appealing to the fear more than to the science? Does that kind of frame your question? Yeah, so this is really the big deal now, is I'd spent a lot of time traveling around talking to scientists about how to talk about science. And really what the whole thing comes down to is, how do we not do the mistake that we've been making traditionally as scientists, which is beat you over the head with science. And really what we need to do is remember that these are concerned people who are really worried about their health and worried about their food. And I love that. And how do we help them understand the science in a very compassionate way as teachers to help them understand? And I think that that's really the way that we can help move the middle. It's going to take a little time, but it's it also could be something that when we have a major breakthrough, like if we do implement the beta-carotene bananas, cassava, or rice, and it changes blindness in a region, I think that's where all of this changes. Yeah. Yes?
Kind of a follow-up to the question that just got asked, but I grew up on a fourth-generation family farm in Iowa. I lived and breathed agriculture my entire life, and some of the people in this room can say the same thing. And I never thought growing up that actually the gentleman that got left out of this room ran into me at the state fair. We had a nice hour-long discussion with him attacking the way of life that I've known for my entire life. And you as a scientist are in the same position, and I think that the thing that agriculturalists and scientists, who are really the same thing, run into is the fact that we have a full-time job in doing what we have to do, and then also on top of that, we have to go on and advocate for ourselves to explain why we do what we do. And I think that that's a very important piece, and I think that that's something that we all have to do. But the time factor is a problem. So first off, I appreciate you coming here tonight and taking your time to talk about something you're passionate about. I really do appreciate that. And then along the lines of just how we go about doing that, how we continue to show not only on my aspect the production part of it, the actual implementation, but the research and scientific part that you're involved in as well.
Okay, excellent question, and I should have shown a slide just for you tonight. One way that farmers, and I speak to tons of farmers, because they're all very interested in, they do feel that their choices are being made by people who don't know the science, and that people want to tell them what to grow. And so they're very concerned about this. And I speak to a lot of farmers in Canada in the off-season about this, even in the States. One of the most important things you can do as somebody who understands this technology is participate in the public dialogue. That when there's a news release about that glyphosate is considered to be a problem, or there's someone is impugning whether BT is poisonous or not, chime in. My family grows it. Here's what I know about it. Here's how it works for us. Here's what we've observed. Here's when we've seen resistance. Here's how we don't get resistance. Your words as a farmer are the most credible words out there when you look at the surveys. Farmers and dietitians and physicians and scientists are right up at the top. But who are the least likely to participate in these conversations? Farmers, dietitians, physicians, and scientists. The easy way for you to start to get involved is to get involved in social media by setting up a website about your family's operation. I don't know if you have a blog that you put this on. You do? Okay, cool. Yeah, look at the Peterson brothers. Look at Brian Scott with The Farmer's Life. Jenny Schmidt. There's so many outstanding farmers who just take pictures of day-to-day operations. They write about it's basically like a diary that we get to look at. And for them, it's no big deal. This is what you've done. This is what we've done for generations. We get to be a fly on the wall, and for many of us, that's pretty exciting. Just to realize that there's a face and a family that's attached to this kind of work, I think helps us really with the message. But keep getting involved in that. Send me your link too, okay? I'll make sure we put it somewhere visible. Yes?
And kind of what I was asking earlier about trying to change society right now, it's a pretty powerful storm against technology. And it seems like that side is really emotional about it. It's rational, they throw expletives,
Bingo. No, it perfectly said. That your audience isn't, Monsanto sucks 666, and you in a chat room. It's the other millions of eyes that are looking at that and weighing who has the credibility here. Yes. People do read those, and I think it's an important thing. That's why it's important. Every place where I've said you're an idiot has been very well screenshotted and come back to bite me time and time again. And you can find things online about me that you just would never believe. I have trouble believing. But really, you have to really keep your cool, because it doesn't change hearts and minds. What changes hearts and minds is talking to people about science and talking about people about farming. If we can just communicate about the realities of agriculture, it goes a long way. And that does mean being sensitive to the potential downsides. I mean, farmers are the best stewards of the land. They don't want to do something dangerous. They don't want to hurt people. It's one of the parts of this I find most preposterous, is that we have 1.5% of the population who allegedly are comfortable manufacturing a toxic compound. I just can't, I can't wrap my head around that. So being very civil in our conversations is good, and I get a little frustrated when I'm here. I probably regret the way I approached him with this. But you get in those moments, you got to really, really watch it. Well, thanks. I think I'd like to call back a few things. We got a little pointy there. Yes. Whatever it is.
She was talking about some.
No, I understand that argument, sure.
And it's like,
Well,
why don't we just have more people out there, I think the emotional part, the emotional appeal that you guys have is this, we're going to stop all these problems in the third world. Because you can't really connect with people emotionally about how they're ready unless you say,
no, I'm with you. Oh, you're right. You're 100%.
So, the emotional appeal you have is we're going to save these people in the third world.
Hmm?
And the fact is, if that's really a way to do, the goal, then there are a lot of other ways that might be a little bit better.
Well, what are you waiting for?
People and communities.
But what are we waiting for then? What are people waiting for? If you have other solutions.
That's the problem. You guys can afford to be civil because all of these companies have so much money. They make so much money off this technology. You talk about these poor little
Yes.
NGOs getting rich off of, this anti-GMO fervor. That's ridiculous.
Well, I'll tell you, Monsanto does make a lot of money, and so does Whole Foods. They're about the same in terms of value on the stock market. The Whole Foods the market. When you look in terms of companies and wealth, and that's a whole nother discussion. I think we should do a lot more for the developing world than we do. A lot of this is very regional specific, that the local skirmishes and warlords and gatekeepers that are involved in local areas are a tremendous problem that we have to bypass. Are there other mechanisms that we could use, like supplementation? It's been tried and it works in many places. But installing a simple gene that comes from a plant into a culturally acceptable plant variety would be a really good solution in some cases. I got a good question for you. Can I ask you a question? What would be, is there a case by adding a gene to something would be acceptable to you? Like by adding a gene by a transgene to solve a problem? You don't have to answer. I just always like to ask that.
I'm just a hobby farmer.
I got you.
I look at it from the holistic perspective, not consumer perspective. I mean, I look at it from the whole thing, like the farmer, the people, the structure of the economy that it's going into. If it makes it a more industrial economy by by creating this crop, then I'm not for it. I think I'm for subsistence farming. I think that's the healthiest way for people to live and eat on the land. I think it's healthiest for the land, and I just don't believe in industrial farming.
Yeah, okay.
I
Well,
don't think industrial farming is the solution for.
what if my GMO strawberries from my lab that don't require fungicide and fungicide, would that be okay? They don't change anything about the plant. They just make it so bacteria fungus doesn't kill it as fast. And that's a pretty good idea, and it's really good for our farmers. That would be fantastic. My industry would love that, but we could never deregulate it. It costs too much. But you see, there are applications that if you let a guy like me play in that space, work with you, we could do good things together. Sure.
With your situation, is probably I would support what you're doing with the orange trees because of the
Well, thank you.
breeding.
I understand.
People have the opportunity to not ingest these things if they don't want to.
Yeah, and
People have conducted this experiment over and over again where they put an ear of genetically modified corn out and they put an ear of non-GMO corn out and the wild animals go straight to the non-GMO.
Except when they don't. I'll tell you what. I've got 500 ears of corn. Half of them GMO, half of them non-GMO, and we are going to do the experiment with school kids all across the country. And in every preliminary trial, we don't know which one's which because they're coded, so it's a double-blind test. But every test we've done, both of them get consumed like that. Nature doesn't really care; they just hork that stuff right down. But would you participate in the study if we send you the corn cobs? Okay, cool. Yeah, we just asked for a picture before.
Because I know somebody who's done the study here in Iowa.
Okay, well, come get my card afterwards. We're going to do 500, it's 1,000 ears of corn. 500 different kids, or other people like you and me, are going to do the test, and we're going to put all the results on a website. We're going to do a big paper with them. Yeah, no, I think it's totally cool. And it's double blind. We have no idea what we got. They had to cut the ears of corn because the non-GM ones were all eaten by earworms on the ends. So they had to cut off the part that would tell which one's genetically modified and which one's not. So we have these short little stubbies because some of them were gone. It would have given it away. Yes?
You talked about using GMOs to have beta-carotene. What other vitamins could we add to plants?
Yeah, thank you. Very good question. So what other vitamins can we add to plants? You can add folate. And it's been done. The lab next to mine made tomatoes that have 35 times the normal folate. Folate, as you know, is the B vitamin that's responsible for assisting an enzyme during neural tube formation. So a number of neural tube defects are caused by low folate levels. And you can change one enzyme called GTP cyclohydrolase, which is the first committed step to creating more folate, one of the subunits of folate. And you can do that and make vitamin-rich vegetables. Folate is also a very important nutrient that crashes in accumulation once you harvest the fruit or vegetable. So you take the strawberry off, its folate levels are high, within 24 hours they're low. My lab is working on ways that you can add light to a treatment after you pick a strawberry to keep those folate levels higher. So that's something that we're working on. That's a good example. Vitamin C also, yes?
But aren't there a lot of vitamins that people say you shouldn't take a whole lot of them, like iron? I know for sure.
Sure.
Folate and iron are very connected. So, I mean, having a ton of a lot of one vitamin, that may not necessarily be a good thing.
Well, you're absolutely right. There's a threshold for danger for everything. But in cases of... No, I'm with you.
Eating food that has 10 times or 50 times the norm, that's what I want to do with.
Well, you got to be really careful with your leafy greens because they got lots of folate in them. And so to be able to make something that would be a regional fruit or vegetable that would have more vitamins could be a potentially very good thing. Of Of course, there's thresholds when it is effective and dangerous, and we have to be very careful to operate in those ranges. But that's stuff that's easily testable. Wow. Yeah, so lots of people hanging on. Yeah, there's one way up on top. Hi. Up in the cheap seats.
Economically, what have you seen for GMOs improving, I guess, the economics?
Well, that's a really tough question. So the question was, economically, what have you seen GMOs improving? And I think that maybe the best testament for where they've been actually used and deployed has been in these big giant ag crops. And the reason that they're still used is because they work for farmers that use them. A lot of the farmers I talk to can't wait to get their hands on the next hybrid that's got stacked traits. They're always looking for the newest and best, and it helps them farm by cutting different farm inputs. If you don't have to use insecticide, you save money. If you don't have to use tillage to get rid of weeds or other herbicides, it saves you money. So that's kind of the advantage. It has to be cost effective for farmers. And when people say, well, it's all about making money for seed companies, it's all about making money for farmers. And those are the folks at the end of the day that if they're not making a buck using this technology, it costs them a lot more to use it. They're not going to continue to use it. So, yes?
Yeah, we're seeing wholesale disasters with the farm group.
Mm-hmm.
Basically, we do to protect their people. We have a IP every 5 years because of the presence of the SEMA and the dairy laws around. So most farmers right now tell their economic times cannot afford those. We're actually seeing higher yields with either lower traded or conventional the last 5 years. In fact, Iowa State is publishing 6-county graph on corn yields and soybean yields in the last 10 years. Corn yields have dropped roughly 15 bushels an acre and soybeans 8 bushels an acre.
Yeah, and I can't speak to the specifics for Iowa, but you're certainly right. The corn rootworm problems have become more severe, and it's definitely a problem. Corn rootworm is always an awful problem and is not easily controlled by many mechanisms. Even crop rotation, the thing is not easily suppressed.
Yeah, the Chinese did a study working with Dr. Bruce DeVosh, and they did a 37-generation study, and they found out the resistance was controlled by a non-recessive trait. The seed companies always assumed if any resistance showed up, it would be controlled by a recessive trait. So the original assumption was wrong.
And that could be. I don't know that literature. I mean, I know of it, but I don't know it intimately. I haven't read through that. I'm not a big rootworm literature guy. I'll have to get into that. I'd be happy to give you a card and we can follow up because I'd love to know more about it. It's not a surprise when you find you talk about recessive traits that would lead to the resistance, or it usually would be something that would be a dominant gain of function mutation that would allow you to metabolize some sort of preventative compound or live through some sort of whatever that protection is. It happens. Nature evolves fast, and no matter what we try to do, we're always in a battle with organisms like bugs and microbes that can generate generations on a much faster time than we can come up with new science. That'll always be a problem.
I think that's one of the issues is the arrogance of the technology, that we can do this and there's just not been a lot of study about its effects on the environment or these types of things.
Well, but
the studies that are done are very minimal.
well, I think I'm not so comfortable with that.
We can solve these problems.
We do solve problems, but every problem is a patch. I mean, every disease we solve in human beings is just opening the door for another one. All of our solutions are temporary. Look at antibiotic-resistant bacteria in human disease that come about from the use of antibiotic drugs and silver bullet technologies. All these things are just the way we can use technology as best we can to help improve the human condition. And there needs to be a lot of participation in that. Hey, I'm going to give you a card.
Oh, yeah, that's right.
Come here.
I'm going to take it over to the book.
Thank you very much for all your questions. I appreciate you a lot. Thank you. Any other thoughts or questions? Are we getting there? Yeah, hey.
Can you tell us more about your research on LED lights on plant traits?
Well, all right, so this is getting back to my research. Our research for years has been how do you not just how do you grow, so I've always been interested in the way that plants interact with light and how light is used as a signal to teach a plant to do things like flower or potentially develop disease resistance or change flavors. And so what we're doing in the laboratory now is using light as a vocabulary of signals in ways that never occur in nature. Talk about freaky, weird stuff. We're using light pulses in different combinations between different ends of the spectrum that can be visual, seen or not seen, so UV and far red light, to customize the way genes are expressed in ways they've never been expressed in a natural context, but give us a very favorable outcome, such as improved nutrition profiles, maybe better flavors, maybe resistance to disease or better shelf life. And those are things that we're doing just with light, which is really cool because we can use a treatment that doesn't involve chemicals, really cheap to apply, and get a benefit for the consumer. So this is a really exciting new technology. I'm glad to be piloting that stuff. You'll see lots of that in the next few years. We're getting there. Oh, I made it. My voice lasted the whole night. If you have any other follow-up questions, I'd be really happy to talk to you anytime. Just send me an email. I'm always pretty accessible. Most of all, I really appreciate you sticking around tonight. It was really nice of all of you to do that. So thank you very much and have a really good night. Thank you.